Hello students, today we continue that gametogenesis. Last class we introduced that gametogenesis. Now we discuss about that spermatogenesis and in that one. In that two types are there. That is, one is the spermatogenesis and another is the oogenesis. First one, spermatogenesis. In this one, who is the scientist? What is meant by spermatogenesis? That means the production of these sperms or production of the haploid male gametes from diploid mother cells and that is we call as spermatogenesis. Haploid formation of haploid male gametes, that means sperm from diploid mother cells, diploid mother cells from testis, that means male gonad. Then that is the process we call as spermatogenesis. Then who is the scientist who coined the name as sperm and all? That is D. Gray and all. So D. Gray coined the term that is spermatozoa. Then who is the scientist who discovered that male spermatozoa? Who discovered the male sperms that is related to human male gamete? That means sperm discovered Anthony Leeuwenhoek. Anthony van Leeuwenhoek discovered that male gamete, that is a human male gamete. In this one, there are particular phases are there. What is the first phase? First phase, multiplication. Multiplication phase. Then this is the first one. Second phase, growth phase. The third phase, maturation phase. The first one, multiplication phase. So already we said what is the common cells which undergoes that particular gametogenesis and primordial germ cells, where it is present inside the seminiferous tuber. So here only all multiplication, growth, maturation phase, it involves the primary sex organ, nothing but testis only. So in this the first phase related to multiplication. In multiplication phase, during this one, primordial germ cells, that is in germ cells, that means primordial germ cells, through mitotic division, through multiple mitotic division, they form into the spermatogonia. Here, in multiplication phase, this is the primordial germ cells. Normally diploid, that means 46 number of chromosomes. These primordial germ cells through multiplication with the help of mitotic cell division. So that is multiplication through. So this cell division is important. Multiplication through mitotic cell division. Mitotic cell division, it forms into the spermatogonia. It forms spermatogonia. These cells we call as spermatogonia. What is the ploidy of spermatogonia? Diploid. What is the number of chromosomes? 46. It is also diploid. The number of chromosomes 46. But there are two types of spermatogonia. Spermatogonia A, spermatogonia B. There are two types of spermatogonia. it forms during that particular multiplication phase and all. There are two types are present. One is the spermatogonia A. The second one, that is spermatogonia B. Spermatogonium related to plural, spermatogonium singular, spermatogonia plural and all. That means spermatogonia or spermatogonium A, spermatogonia or gonium B, like that two separately it will present. Which one undergoes spermatogenesis process? Either spermatogonia A or spermatogonia B. Spermatogonia B undergoes the further that particular growth as well as maturation phase and all. Among this one, spermatogonia B, it undergoes growth as well as maturation phase and formation of that male gametes and all. Then what about spermatogonia A and it acts as the stem cells. Why? Because again ready to formation of one more type of spermatogonia in next type for multiplication, for next type for division, for next type for the formation of that particular male gametes and all. So spermatogonia A, it acts as stem cell. This stem cell again after multiplication again formation of spermatogonia only. So spermatogonia A after mitotic division again formation of spermatogonia only. Whereas spermatogonia B further formation, that means growth maturation phase, it involves and formation of the male gametes and all. Then what among this one, spermatogonia B, suppose this is the B and this is the A. That B, spermatogonia B, it acts as growth phase and all. It undergoes growth phase. This spermatogonia B, the same spermatogonia, it proliferate or it increase the size with the help of nursing. Already I said, Sertoli's provide the specific nutrient food material for growth and development or maturation or synthesis of these particular gametes and all. So with the help of Sertoli, it increase the size. It increase the size and formation of primary spermatocyte. Little bit increase the size. That is we call as primary spermatocyte. To increase the size, primary spermatocyte with the help of nursing. Then this primary spermatocyte, what is the ploidy of primary spermatocyte? Many times in examination they ask what is the ploidy or what is the number of chromosomes present in primary spermatocytes during spermatogenesis. Then what is your answer? It is actually diploid condition. Primary spermatocyte, the number of chromosomes 46. That means it is a diploid condition. Why? Because same cell only it increase the size by nourishing with the help of Sertoli cells. Then this primary spermatocyte now original it undergoes meiosis cell division. This originally it undergoes to the meiosis. Meiosis 1. So this is the growth phase. Growth phase means what I understood. Multiplication phase means there is a primordial germ cells due to multiple mitotic cell division, formation of spermatogonia we call as multiplication phase. Then when it begins, when it starts at the age of puberty. What is the puberty age? That is 13 to 16 years. That is the puberty age and all. In that particular period of time only it begins and all. Then in this two types of spermatogonia are formed. One act as a stem cell, again it ready to formation of the same type of cells and all. Then another type, that is the spermatogonia B, it undergoes further growth and maturation phase and all. Then it forms into the primary spermatocytes and all. These primary spermatocytes with the help of Sertoli cells it nourishes, then increase the size and all. Then it is also same diploid condition. Then originally it undergoes reduction division. Originally it undergoes meiosis cell division and all. From here only it ready to start maturation phase. So maturation phase, nothing but reduction cell division. Maturation phase means simply remember reduction division. Here primary spermatocyte after meiosis 1, it will be forms into the two haploid cells equal, that is it forms into the secondary spermatocytes. That means the equal size, equal number of the chromosomes of the secondary spermatocytes. Two equal and functional two spermatocytes are, that is important. Two equal as well as functional secondary spermatocytes are present. What is the ploid here? Here meiosis 1 takes place. Meiosis 1 means it reduces half of its parental chromosome. What is the parental chromosome here? 46. What is the number of chromosomes present in secondary spermatocytes? That means 23 each and all. This is the 23. This is the 23. So it represents as this one haploid 1. It is related to haploid number of chromosomes and all. Secondary spermatocyte, immediately it undergoes meiosis 2 cell division and all. These secondary spermatocytes, here I have written, right here, secondary. These are the secondary spermatocytes. It is the haploid condition, each and every one 23, each number of the chromosomes and all. Then it undergoes meiosis 2 cell division. This is, and this one. It undergoes meiosis 2. That means reduction cell division 2. Already you know, in first year also you studied, meiosis 2 is equivalent to mitosis. Whatever the number of parental chromosomes, same number of chromosomes only it comes to the daughter cells and all. So that's why it will be formed into the 4 equal, the functional, it will be formed each and every 23 each number of the, it will be formed into the spermatids are formed and all. These particular 4 we call as, this is spermatids and all. But all are functional, that is most important. All 4 equal functional gametes are formed, that is we call as immature gametes. All equal, 4 equal haploid, that functional, that is immature gametes are formed, that is we call as spermatids. What is the number of chromosomes present each and every spermatid? That is the 23 each and every one now. It is the immature one. So what we call as formation of spermatids from spermatogonia. Formation of a spermatid from spermatogonia is called as spermatogenesis. What we call as spermatogenesis and all. Then that immature spermatid, then it transfer into the mature sperm and all. Immature, it will be transferred into the mature sperms and all. That is immature, it will be transferred that mature sperms and all. It will transfer mature sperms. This is the sperms. So it will transfer spermatid transfer and formation of a sperm. Then what we call as, that is this process we call as spermiogenesis. So remember carefully, spermiogenesis. That is spermiogenesis or spermatogenesis and all, or spermiogenesis. Spermiogenesis means transfer of immature spermatid to that mature spermatozoon. That process we call as spermiogenesis. Spermiogenesis means that spermatid to sperm. That is, what is known as spermatogenesis? Spermatogonia to. That is, what is meant by spermatogenesis? Spermatogenesis, that is spermatogonia to spermatid. That is we call as spermatogenesis. Then, after formation of this sperm, immediately here different parts are present, head is present, neck is present, tail is present, and this part is present. In that matured sperm, it embedded on Sertoli cells and all. Then immediately it embedded on Sertoli cells. That sperms, it is present on Sertoli cells. The head of these sperms, it embedded on Sertoli cells. After a certain period of time, it is separated from Sertoli cell to the seminiferous tuber. Then what we call separation of mature sperm from Sertoli cells to seminiferous tubule. That process we call as spermiation. Then separation of here, that spermiation, that means separation of spores from Sertoli cells. That is we call as spermiation and all. So already said which type of the hormones it must be controlled. That is a follicle stimulating hormone which control the Sertoli cells. Whereas LH hormone to control to the leading cell for secretion of androgen for complete formation, that maturation of these particular spermatogenesis and all. So both LH as well as FSH both act as a crucial role for this spermatogenesis, which are controlled by the anterior lobe of pituitary gland. Anterior lobe of pituitary gland controlled to the gonadotropin releasing hormone and also gonadotropin releasing hormone from hypothalamus. It stimulates to the anterior lobe of pituitary gland. Then it secretes FSH and LH. FSH act on the particular Sertoli cells, whereas leading cells it must be act on that is specific to interstitial cells or leading cells and all. There it helpful for secretion, releasing of the particular hormones and all, controlling the particular process and all. So here in this one, an averagely how many number of sperms are released in healthy human beings and all. The healthy human being it will be releases 200 to 300 million. So both testes 200 to 300 millions for one ejaculation. Only one ejaculation, 200 to 300 millions of sperms per day. It will be releases and all. It has one ejaculation will be present 200 to 300. In that one 60% normal and average and all. 40% vigorous. Why? Because male gametes, that means the spermatozoans, has present microscopic motile nature and all. Microscopic minute motile nature. Because of the motile nature, it will be present under here 60% average. Average, that means a normal motility, not vigorous and all. Whereas 40% it will be present that vigorous motility, vigorously ready to remain 60% unable. So the person, whenever, why? Because these for complete maturation of this sperm, it is also needed the axillary glands like the seminal vesicles, then which are present in epididymis, axillary tubes and all, as well as vas deferens. While moving that spermatozoa all along with the axillary tubes and all, they are also it ready to mature. They are also it ready to perfect. They are also it ready to increase that motility and all. They inside the epididymis, that particular lifespan of the spermatozoa, that is 24, that is 4 weeks. It will be ready to, that means 1 month, it is ready to alive and all, related inside the epididymis. Whenever after ejaculation, The male gamete ejaculated from male reproductive system to female. If it is enter into the female, the lifespan of the male gamete 48 to 72 hours only. That means 2 to 3 days maximum. Within that, if it is available female gamete, then ready to fertilization. Otherwise, it may not take place any type of one. Suppose less than 20 millions per ejaculation. Why? Because one ejaculation 2 to 3 ml. One ejaculation 2 to 3 ml is present under ml of the semen it secretes under 2 to 3 ml of the semen it must be released. What is the semen? Seminal plasma along with sperm. Seminal plasma along with sperm. That is we call as semen. That is equal to seminal plasma plus sperms. That is we call as semen and all. Per day 2 to 3 ml. So at one ejaculation is present 2 to 3 ml. 1 ml 100 millions are present. So 2 to 3 ml it will be present. 2 ml per ejaculation. 1 ml it is equal to 100 millions. Millions of sperms. 100 millions of sperms. Suppose if it is present less than 20 millions, that is we call as oligospermia. Whenever it is present less than 20 million sperms, one ejaculation, such type of person we call as, that is such type of disorder, that is we call as oligospermia. Suppose ejaculation semen is there, but completely absence of sperms. Seminal plasma only present, sperms are completely absent. Completely absence of sperm in seminal plasma in semen. That is called as agiospermia. Agiospermia. So any one among the defect in male, whether it is oligospermia, whether it is agiospermia, that may causes infertility of the male and all. So if whenever increase more than 20, more than 50 at least, then ready to start to the fertility, ready to start to the fertilization and all. That is to avoid that sterility and all. So agiospermia means completely absence of sperms. Only semen is present. Semen means seminal plasma along with sperm. Completely absence. Any one among the genetical defect or any one among the effect and all. That is agiospermia, completely absence of the sperms and all. So each and every sperm it will be present. 3 particular parts are present. That is, if we observe, suppose this is that is we call as, that is normally we call as spermatid and all. How it will be developed spermatid to spermatozoa. In spermatid, this is we call as spermatid. In spermatid, this is the nucleus. This is the Golgi complex. This is the mitochondria. These are centriole. Then this is spermatid. This is the nucleus, Golgi apparatus, and mitochondria, and one among these one centrioles, proximal centriole, distal centriole and all. One among the distal centriole, it continues as this one, that particular axonium and all. Then in this one, it continues, little bit enlarged. So the Golgi apparatus, it modifies and continues regarding related to the superior part of the head, that is the acrosome and all. Then here, then mitochondria, everything is present. Then further it forms. These are mitochondria. This is the centrioles, the axonium. This is the acrosome. This This is the head. This total that is related to nucleus. Actually, the nucleolus may not be appeared, only it will be present. Nucleus is present, nucleolus. It completely absent central. Here it will be observed only chromatin fibers. That means chromosome. What is the number of chromosomes? It is present 23 under each and every. One among this one related to negative. That means X chromosome. Another one Y chromosome. So 50% of the chromosome, suppose 20 to 200 million sperms are released under. In that 100 million Y chromosome, that is androspermia. Remaining 101, it will be present to X chromosome, that is gynospermia. So gynospermia related to X chromosome containing one, androspermia Y chromosome containing another. So 50% Y chromosome containing of the spermatozoals. Remaining 50% related to X chromosome under. So gynecospermia related to X chromosome, androspermia related to Y chromosome. Here Golgi apparatus, it modifies into this one, acrosome. So after complete maturation, if you observe, after complete maturation of the spermatozoon, it is present like this manner, one head part as well as middle. Why? Because centrioles, in this spermatid, observe carefully, in spermatid Golgi apparatus, it modifies into the acrosome. Then nucleus, it covered throughout the head part and all. Then centrioles, it forms related to that axoneme, that means the tail part, one among these one. And one among the centriole, it forms into the neck part and all. Then the mitochondria, which occupies to the middle part and all. So actually the mature spermatozoon, the structure, if you observe the structure of spermatozoon, one is the head part is present, then middle piece is present. Suppose this is the head part, this is the head, this is the neck part, this is the middle piece, and this is the tail part. And this is the tail part. The superior part of the head, it will be present related to crown-like structure, cap-like structure. That is we called as acrosome. Acrosome contains specific enzyme. What is the name of that enzyme? Hyaluronidase. It contains hyaluronidase. So it acts as vital role for penetration of ovum and all. So which part of, suppose related to spermatozoon is there, acrosome will be absent. Apart from this one, acrosin, otherwise we call as acrosin, hyaluronidase. These are the enzymes act as a vital role to penetrate into the female gamete and all. Then in this middle of this one, maximum. So maximum it covers related to that nucleus and all. Maximum it covers into the nucleus. So this is the head part, superior head part and all. Then related to, in this particular, this is the proximal centriole and this is the distal centriole. It is the proximal centriole. This is the distal centriole. Then another very important, the distal centriole, which continues as this one axoneme, the distal centriole, it continues as this one, this part we call as axoneme. But here the basal part, one more centriole is present, the annual or round centriole, but there is no specific function related to round or centriole related to that particular part and all. Then this is the middle piece or middle part and all. Related to middle part and all, throughout this one plasma membrane it covers throughout this cell and in this. Then in that middle piece normally it contains mitochondria. That is these mitochondria. That's why it is called as a powerhouse. It helps to enhance the motility of this sperms and all. But it is spiral one another. These ones spiral one another towards the axonium. It is spiral one another towards the axonium. Axonium normally it contains whatever axonium, that is the 9 plus 2 arrangement. It forms into the specific flagellum to change the direction of the sperms and all. So middle part of the spermatozoa act as an important role because of the spiral mitochondria which coil towards the axonium. Whatever spiral mitochondria which coil towards the axonium, this is we call as Nebenker. This particular process or this particular involvement of this one we call as Nebenker. So this is the total. What is the Nebenker? Nebenker nothing but spiral mitochondria which coil all along with the axonium. That is we call as Nebenker. Then here the posterior end. Whatever here the posterior, half of the posterior here, half of the posterior and the neck part as well as along with middle piece. Half of the posterior, neck and middle. Not tail part. This is the head, it is the middle part, this is the tail part. This is the end piece. That is the end piece, this is the main piece and all. Below this in the tail, this is we call as the main piece. That is the end piece and all. Half of this one it covered with the specific sheath. That is we called as manchette. What is the name of this particular part and all? This is we called as manchette. Whatever half of this one which it will be covering and all. The proximal centriole acts as a vital role, the first cleavaging of the zygote and all. So, proximal centriole vital role for cleavaging of first zygote. Then, distal means it continues as this one axonium and formation of flagellum. That axonium nothing but it formation of flagellum. Flagella only it acts as the tail for changing the direction and the main piece it covered with the fibrous sheath and all. The main piece, this is from here to here, it covered with fibrous sheath. It covered with fibrous sheath and all. The main piece covered with total that related to fibrous sheath. It covered with fibrous sheath up to there. The end piece terminally, it is not at all covering and all. So, head part, acrosome, vital role, act as important role penetration of female gamete and all. That neck part, 2 centrioles are present, proximal centriole act as a vital role for first cleavaging of zygote and all. The distal centriole, it continues as axonium and formation of that specific part. The formation regarding related to axonium, that means flagellum. Flagellum will be present under, then this actually the round centriole may not be present here. The round centriole it will be present here. This is actually the round centriole. The round centriole originally present the end part of the middle part and there is no particular function, still not known. What is the specific function, or there is no specific function of the round centriole or annulata. Actually, it will be present here, the last part of the middle part. And what is the important role of middle part? Due to presence of spiral mitochondria, it covered all along with axoneme, that's why we call it as Neben curve, it continues. But tail which change the direction, rotation of this one. But end piece, it is not covered anything at all. So acrosome, it covered completely due to presence of higher lunaridase as well as acrosome to penetrate on this. It is a minute microscopic. Just now I said, inside the epididymis, 4 weeks it must be living under, but whereas once it expelled, then automatically where it will be reaches whenever reaching to the female reproductive system under in female genital tract, it is survival 48 to 72 hours under, but it is a motility in nature. So based on this spermatogenesis, many times they ask from where the spermatogenesis, from where which type of this cell act as a parental for formation of the male gametes under. Then you write spermatogonia. Then what is the ploidy of primary spermatocyte? The ploidy of primary spermatocyte is the diploid. That is, what is the number of chromosome? 46 under. What is the ploidy of secondary spermatocyte? Haploid. Why? Because after meiosis, one division under. Here, all are functional, whatever the spermatozoa. But every day, whenever begins for puberty until old age, the testis has capacity to produce the male gametes under. Then, what is it by semen? Along with the seminal plasma plus sperm. That is we called as semen under. That is related to semen under. Then, which hormones control the spermatogenesis? That means gonadotropin releasing, which is secreted by the hypothalamus, then it stimulates anterior lobe of pituitary gland and it releases FSH. FSH acts as Sertoli. Sertoli already you know particular factors are releasing. What are the factors? Androgen binding protein as well as inhibin under. Then apart from this one, LH hormone, it stimulates the Leydig cell. Leydig cell after stimulation, secretion and synthesis and secretion of androgen. That means testosterone. It is also act as a vital role for the completion of the process and all, as well as secondary sexual characters related to testosterone. Whenever absence of testosterone, there is also one among the sterility of the male and all. That is, what is the number it releases for ejaculation? 200 to 300 million and all. 50%, that is the Y chromosome contained, that is the androspermia. Remaining 50% gynospermia, that is X chromosome containing and all. So, that is, this is the complete structure of this, the specific human spermatozoa. This is the structure of sperm. Complete maturation. Another many times they ask what is done by spermatogenesis, spermatogonia to formation of spermatid, we call it spermatogenesis. What is done by spermiation? Spermatid to transfer of spermatozoa. That is we call it as spermiation. What is done by? That is the spermiogenesis. So spermatogenesis, spermatogonia to as well as spermatid. That is spermatogenesis. What is done by spermiogenesis? That is the spermatid transfer into spermatozoa. That is we call it as spermiogenesis. Then what is the spermiation, separation and removal of sperm from Sertoli cell to the seminiferous tuber. That is we call it as spermiation. So these are very very important in exam point of view. Remember carefully, diagrammatically, observe, then understood completely. Okay. Remaining we discuss next class.